Welcome to Brain Ponderings, your window into the world of neuroscience research, conversations with experts in their particular area. And today it's my great pleasure to welcome a friend and actually a lab member a long time ago, Norm Haughey, who was a longtime professor at Johns Hopkins University in Neurology Department, and he recently moved down to New Orleans to Tulane, which is very exciting to get new environment, new lab, more energy into the research program. And Norm has done a lot of work in different areas, and I'll let him talk about it, his trajectory of his research. But what I want to focus on today is certain lipids in the membranes of cells, sphingolipids, and some lipids derived from them through enzymatic activity, ceramides. And work he's done and kind of the field's done on these particular lipids and their roles in neurodegenerative disorders mainly is what Norm's focused on. So welcome, Norm.
It is. Thank you, Mark. It's a pleasure to be here, and it's very nice to see you again.
Yeah, likewise. It's been a while, quite a while.
It has been a while. It has been a while. So the story how I trained with Dr. Matson here as a postdoctoral fellow I was doing my PhD in Winnipeg at the time, and my advisor Jonathan Geiger decided to go do a sabbatical with you in Kentucky. You were at the Center on Aging in Lexington, Kentucky at the time. And while he was gone, I had secured a postdoctoral position with a guy named Stuart Lipton at Harvard. And Jonathan came back and he said, no, Jonathan Geiger, who was my advisor, came back and he said, you got to go work with Mark. And so I started looking into what you were doing. I think I had a 10-minute phone conversation with you. And next thing I knew, I was in Kentucky. And it was the best decision I've ever made.
Okay, well, it's good to know that Jonathan recommended my lab after he was in the lab.
He still tells me that that was the most productive grant writing time of his life,
Uh,
was the sabbatical that he did with you. Just all kinds of new ideas floating around freely within your group, and that was my
uh,
experience as well.
that's true. My lab's always been it's focused but not focused. I don't know if that makes any sense, but
Mm,
I always like to think broadly and try to incorporate new ideas and
yeah.
models and kind of broaden the field, get an idea of the big picture.
And then the idea to work on sphingolipid metabolism came from a guy in your group named Roy Cutler.
Yeah.
Remember him? And
Oh yeah.
at the time, he was telling me about a small company that he had developed using mass spectrometers to measure trace elements in the blood of athletes, wealthy people who really want to dial their health in as much as they possibly could. And one of the products one of the metabolites that they were very interested in were sphingolipids. And he talked me into taking a look at that time at a group of people infected with HIV, looking to see if there were changes in circulating sphingolipids, and and that started a 20-year career of sphingolipid metabolism.
Yeah, and he'd use a method mass spectrometry to identify and kind of quantify the single lipids and ceramides, these lipid membrane lipids in tissue from, well, he actually had tissue from humans
Hmm.
of different ages, brain tissue, and then also some Alzheimer's disease
Yep, I
patients.
heard.
It had a nice PNAS paper on that.
I remember that. The first paper on Alzheimer's disease came from your group, and the first HIV paper came from my group. They were about 2 years apart.
Yeah.
And
Uh,
you were of course on both of them. Um, I think at the time, so without mass spectrometry, the way people used to look at lipids and the various metabolites of lipids was thin layer chromatography. It was one of the main methods, which it doesn't allow you to identify any subspecies. And of course, um, Different lipids have different chain lengths. Those chain lengths and the degrees of saturation matter a great deal in terms of their biological function, and you just couldn't dissect any of that. And then, 20 some odd years ago, mass spectrometry came along, and all of a sudden, people were able to identify thousands of different lipid subspecies. And that sort of began the revolution of being able to understand the biological importance of these and the metabolism. So without mass spectrometry, it never would have moved forward.
So when you got the faculty position at Hopkins, then I guess one of the first instruments you got was mass spec.
It was for many, as soon as I could afford one. They are not cheap instruments, you know.
Yeah.
So for years we collaborated with you because
Yeah.
Roy and yourself are very good collaborators, and we used your mass spectrometry until I could afford one,
Yeah.
and we've had at least one, if not two, of those instruments in the lab ever since. Um,
Do you want to explain a little bit more about what sphingolipids are and where in the membrane of cells they are? They're not necessarily distributed uniformly and kind of a background on these lipids.
Absolutely, absolutely. They are a class of phospholipid with very unique properties. So they are highly saturated, which means their tails, they have two fatty acid components, or two carbon chains, I should say, and they tend to be more saturated than phospholipids. So phospholipids have a lot of unsaturated bonds, which means their tails, I'm just going to use my hands, their tails have a kink, right? And the more number of unsaturated bonds, the bigger the kink. And that does not allow them to pack together very tightly. They're much more fluid. Sphingolipids, because they do not have a lot of unsaturated bonds, their backbone has one or zero, and then the backbone has zero, and then the variable chain has either one or zero unsaturated bonds, and it allows them to pack together very tightly. Um, they can be converted through a number of different enzymes into a product called ceramide, which simply takes off part of the head group of the sphingomyelin, which makes it it creates an ability for them to pack together even tighter. They form less fluid portions of the membrane called lipid rafts or membrane microdomains. And within these microdomains, they serve as signaling hubs. So certain receptors, certain proteins like to exist within these domains for reasons that we are still trying to understand. There doesn't seem to be a sequence per se, an amino acid sequence that allows a protein to exist within this domain or to prefer to exist within this domain. It's more of a three-dimensional configuration. Things like what they call tubby domains, bar domains are also lipid protein binding domains. So these regions and the enzymes that control the metabolism of sphingomyelin to ceramide by removing that head group are very dynamic and very rapid. And what we found over the years is that signaling even through an NMDA receptor, an important excitatory amino acid receptor on neurons, can cause these receptors to cluster. So the sphingomyelin will be converted to ceramide. The ceramide causes these receptors to cluster together. You get a very strong signal, and then they dissipate because the ceramide is metabolized into other products. And so it's a way of clustering signaling and then a way of terminating signaling by removing those lipids and then allowing the receptors to drift apart and internalize. Over the years, we found that most of the receptors, when they exist inside these domains, are very difficult to internalize. So, for instance, an AMPA receptor that normally internalizes very rapidly after being activated will not internalize if it's inside a raft. It's sort of stuck in the membrane. So the machinery to internalize it and recycle it or degrade it seems to exist outside of these domains.
Interesting.
Yes. Uh,
And then there's usually when you think of receptors being internalized, I guess there's several ways, but you can get an invagination of the membrane, I guess, and have it go inside the cell in a vesicle, like, or often these receptors are not necessarily directly, but through intermediary proteins interact with the cytoskeleton, the cell cytoskeleton,
Yeah.
and that may be another way to influence their localization.
And that's another a very interesting property of ceramides is they have, if you think of a biological membrane, and whether it's the plasma membrane or any of the internal organelles, membranes just don't spontaneously curve, right? There has to be a certain lipid composition that allows that curvature to happen while keeping the membrane properties intact, because you don't, if you force a membrane to curve, you're going to have some leakage. It's just not going to be an intact membrane. And one of the things that ceramides do very well is to help form that curvature. Um, and we found that to be very important just recently for HIV budding
Huh.
and for extracellular vesicle biogenesis. So, in order for a retrovirus of any kind, although we're finding some retroviruses are less dependent on an enzyme that creates ceramide called neutral sphingomyelinase. It takes the head group off sphingomyelin, as I mentioned earlier, and turns it into ceramide. So this enzyme gets activated at the time when the viral components assemble at the membrane. We knew that these viral components, these viral proteins, liked to begin to coalesce at lipid rafts, and then that is where the budding process occurs. But up until recently, people hadn't described exactly how budding occurs other than a certain protein called the GAG pole polyprotein causes the membrane to curve. So what we've found so far is that that protein likes to interact with this enzyme, neutral sphingomyelinase 2, and then that allows the membrane to begin to curve. So the virus begins to bud from the cells, and it allows the round shape to form, and then ultimately there's a cleavage process that allows it to seal, and the virus buds.
Right. So
So
so virus isn't transmitted between cells by just like individual virus particles being released from one cell and then getting in another, but rather they're released from a cell in association with these small vesicles.
that seems to be the case. There have been reports of viruses encased within some of these vesicles, sort of a double membrane. And there have been reports, more reports of the virus being released. In this case, we're looking at HIV, and it's associated with a lot of these other small particles being released from cells at the same time. So it's more of a complex, and we're still trying to sort of understand what the purpose is of these other extracellular vesicles that look like viruses. They're the same size as viruses. They have a double membrane. They just lack the protein spikes that different viruses have. GP120 for HIV, or if you want to look at COVID's not a retroviruses, right? But there are certain spike proteins that differentiate the um, type of particle, I guess it is, that you're looking at.
Okay, so then you mentioned this neutral sphingomyelinase enzyme that generates ceramides from sphingomyelin. And so if you genetically or I guess through a drug or chemical inhibit that enzyme, so knock it down, reduce the levels or reduce its enzyme activity, does that prevent transmission of HIV virus from one cell to another?
So we found some very interesting results. We worked with a group while I was at Hopkins for almost 10 years, developing small molecules to target this particular enzyme. Um, we were interested in this particular enzyme because it has some very unique activation properties. It's directly activated by TNF, well, indirectly through a linker. The TNF alpha receptor has a linker to this enzyme, and it generates ceramide, an innate immune response. So certain inflammatory mediators will activate this enzyme. So we started developing small molecules against it. And as you can imagine, it's an enzyme that modifies lipids. So the types of things that like to interact with it are very hydrophobic. So to develop a small molecule that has enough um, aqueous solubility to be
Uh,
so so any enzyme that modifies a lipid component, of course, interacts with lipids. So we screened at that time 40,000 lead compounds with a group at Prague called the IOCB. They developed initially one of the small molecule inhibitors of HIV. Um, and then with NCATS, which is an NIH-based program that has their own library, and then with some proprietary libraries that were owned by our collaborator and at that time a company called Eisai. So out of these 40,000 leads, we came up with two series. One was called DP-TIP, and the other one was called PDDC. Uh, and 6 years of chemistry later, we came up with two molecules that one had an IC50 of 100 nanomolar. The other had an IC50 of 300 nanomolar, which is, within fighting range to sort of
yeah.
develop it. And then we've used these in two different model systems, and I'll talk about them separately. We're talking about HIV now, and the other one is a tauopathy-based model system. But let's begin with HIV. So you can take a humanized mouse, so a mouse that has a partially humanized immune system, and you can infect them with HIV. They'll develop, HIV will continue to replicate. It will get up to a steady state level, and it'll stay there for as long as the mouse is alive. Um, Antiretrovirals are a series of drugs that have been developed to suppress HIV. You need three different antiretrovirals to effectively suppress HIV for a very interesting reason. And the reason is that the reverse transcriptase is not accurate. So somewhere between 1 in 1,000 to 1 in 10,000 copies of HIV are correct, and the rest are mutants. Most are not functional, but of course, now and again, you're going to get a particle that is resistant against drug X. So because it is so inefficient at replicating itself, it gets around drugs. So finally, we figured out that you need three antiretrovirals targeting three different parts of the replication pathway in order to fully suppress this virus. And that, of course, it began in the 1990s. I think we had triple therapy by 1995, and it's worked very well to keep the virus suppressed. It is not a cure by any means. As soon as antiretroviral therapy stops or is paused or people forget their doses, there's a viral rebound, and the virus rebounds extremely quickly. Within days, you can see its replication again.
And you should mention, Norm, right now that you're most interested in the effects of HIV on the brain because of this condition that a lot of HIV patients are developing now.
That is correct. That is correct. We became interested. One of the first papers on the neurological consequences of HIV infection came from my former department chair at Hopkins. Back in the 1980s, they described dementias that became readily apparent in people with HIV. With antiretroviral therapy, those dementias became less severe, but they still have mild to moderate forms of cognitive impairment that collectively are known as HIV-associated neurocognitive disorders. And those disorders, to date, are still present in somewhere between 15 to 30% of patients infected with HIV. And of course, the longer you live, the greater the likelihood that you will eventually develop some form of cognitive impairment. And the other thing about brain is when we look at where antiretrovirals go, many drugs, including antiretrovirals, do not get into brain very well. So the brain has become a reservoir for HIV that is an extremely difficult target, as you know.
Yeah.
Um, so what we found in these humanized mice, we treat them with antiretrovirals, and then we take away we get a suppression, the viral replication goes away, and then when we release that, we take away the drugs, there's a very rapid rebound, same as occurs in humans. With our drug, PDDC, we're able to, with the same trajectory, reduce replicating levels of HIV. But if we go below detectable limits, if we're able to effectively get the replication to at least disappear, we can detect as low as 2 copies of virus. If we can get below that, we didn't see any viral rebound. In other words, these mice, for all essential purposes, were cured. And then we ran them out for months and months and months, and there was no sign of virus in any tissue, any organ, including brain. These drugs we developed, one of the criteria was we wanted to have a decent brain penetrance. Um, and what we're starting to find now is what this drug does is I mentioned how HIV requires this enzyme, neutral sphingomyelinase, in order to bud. So the virus still buds from cells, but it never closes. So it kind of has an open tail to it. It never forms that circle. And by doing that, it never matures. So you end up with a viral product that is not mature, not infectious. Um, and in the cells that are infected, because you're disrupting that process, eventually all the proteins, the viral proteins that it's trying to produce, get stuffed into lysosomes. It's just the cell is simply trying to degrade all these proteins that are being made. It overwhelms the cell and it kills the cell. So we ended up finding a small molecule inhibitor of this enzyme called neutral sphingomyelinase that effectively blocks HIV replication and selectively kills the infected cells. We've
Huh.
got 100 times above the dose in the effective dose for HIV and in normal cells, and it seems to have no toxicity, at least at that level. Um,
And does the and your work up in Canada, right, for your PhD, you were looking at these so-called coat proteins on the HIV virus, TAT in particular, and finding they were neurotoxic, and particularly striking was you found that uh, those proteins, which can apparently be released maybe from the virus, they increase the vulnerability of neurons to excitotoxicity, which we think that form of death damage to neurons is occurring in a lot of neurodegenerative conditions. So does the does the HIV itself affect levels of ceramides in cells? In other words, uh, since the generation of the ceramide seems to be valuable for the virus to be able to bud off and transmit infection between cells, it would be to the advantage of the virus if it could somehow increase sphingomyelinase activity or otherwise increase ceramide level.
and it does. If you look at HIV infected individuals, we've looked at either cerebral spinal fluid, circulating levels in plasma. Ceramides are certainly increased. In infected cells, you are correct. The cell starts increasing the amounts of ceramide, presumably because the virus wants ceramide to be there, because that's where it assembles and that's where it buds from cells. The mechanisms by which that occurs really are still not that well understood. Um, we think it's one of the viral proteins that are interacting with this enzyme, or maybe higher up the pathway. There's a de novo pathway where serine palmitoyltransferase is the rate limiting step. It may be at that point. We're still trying to understand that. But yes, this virus, and in fact, all viruses, create a local environment that is favorable for their own replication, right? They hijack host cell machinery in order to produce things that are favorable for their own replication.
Yeah, yeah.
Oh, yeah. So the other component that really I think now is taking off even more is the associations of ceramides with neurodegenerative disease. And we started some of this work, I think, with you. Um, at that time,
And,
we were
and then
simply,
and then you got together with Kostas Laketsos and Michelle Melky, right?
that's correct. That's correct. She was at Hopkins at the time. Costas is still chair of a psychiatry there. And we started looking just at circulating levels in a number of different cohorts. The Women's Health and Aging Study, the Baltimore Longitudinal Study on Aging. And what we found is that with Alzheimer's, there was increased levels of certain forms of ceramides in brain, in circulation, and in cerebral spinal fluid. This dysregulation got worse with the degree of cognitive impairment. It was associated with vascular disease. There were a lot of interactions with different components of the disease that we found. I think one of the most interesting things we found, we looked at somewhere in the range of 3,000 subjects from the Baltimore Longitudinal Study on Aging who were recruited at 75 years of age, if I no, 65 years of age, and then followed, for decades. And we had repeat samples from these individuals. And what we found was quite striking sex differences where women compared to men, both men and women with age started to accumulate some of these ceramides in circulation. But women, the rate of accumulation was at least double that of men, especially somewhere, around 70 years of age and above. Um, at the time, we didn't really know what to do with that. It was a very interesting phenomenon. And just recently now, we're starting to figure out that the enzymes which control sphingolipid metabolism change differently with age in men than they do with women. So there is two, if you create ceramide and you stop there, as you said, that can be toxic. It can create these lipid raft domains. It can alter signaling processes in ways that are more excitotoxic. But if you continue to metabolize ceramide, you end up with products that are actually quite protective. Sphingosine 1 phosphate, sort of downstream from that.
Mm.
Uh, and the enzymes which control ceramide metabolism go up with age in women, not so much in men. And the enzymes which regulate the downstream more protective products seem to go up in men with age, but not so much in women.
Huh.
So our thinking now is that one of the reasons for the increased susceptibility of women to Alzheimer's disease uh, could be these changes in sphingolipid metabolism that occur quite differently in women versus men. Um, right now, we're trying to rule out the most obvious thing to see if there's any estrogen driven effects that change the expression of these enzymes. I hope we know the answer to that fairly soon.
So in that
Um,
case, you'd think that, after menopause, then right. So then you'd think maybe, I don't know, before menopause, age 50, there's no difference between men and women, and then after menopause, it starts to see that.
that would be the thought. The samples that we have been using are the biggest sample set we had access to. They were recruited at
Oh,
age 65.
yeah, yeah.
Yeah, so unfortunately, we don't know how comparable men and women are at the younger years. Um, there's a gentleman named Ikizu, who I think he was at I think he was at Harvard at the time. He's now at the Mayo Clinic in Jackson. As I mentioned, neutral sphingomyelinase generation of ceramide creates curvature, which is good for the release of viral particles and the release of extracellular vesicles. These are vesicles that are released from cells All cells release these things. Some of them are simply removing garbage, things that can't be degraded. The cell tries to package it and get rid of it. And we're finding that a lot of them carry microRNAs, proteins, small molecules, in some case metabolites. And it's a signaling process, either a signaling to the next cell. We published a nice paper a number of years ago showing that with a local inflammatory insult in brain, these vesicles are released from brain. They go all the way to liver, and they turn on PPAR machinery that ultimately activates neutrophils that go to brain. So the same could occur
Huh.
in Alzheimer's disease and to spread tauopathies. So this gentleman found that tauopathies begin in the entorhinal cortex, right? And then there's a very orchestrated spread throughout the brain. And he found that that spread of a hyperphosphorylated tau protein that forms these neurofibrillary tangles is through extracellular vesicle release.
You know
Um,
that initial finding, this is interesting historically.
hmm,
So I got my PhD from University of Iowa.
I remember.
And when I was there, there was a guy named Brad Hyman
Hmm,
who was working. He was across the river. I was on the liberal arts side. He was on the medical center. And it was there at the University of Iowa that they described this entorhinal cortex as where the tau pathology first began, based on this
yeah.
postmortem histology and so on. And so maybe that's connection to Harvard because Brad Hyman's been at Harvard since then, I think.
Oh, that could be. That could
Yeah.
be.
Yeah.
Um, so he did some very elegant experiments to show that extracellular vesicles were involved in the spread, and that if you block extracellular vesicle biogenesis, you essentially halt the spread through a number of different model systems that he developed. And so we came along with our small molecule and contacted him and began looking at if we inhibit neutral sphingomyelinase, what do we do to the spread of tau? And this small molecule does a fantastic job, very similar to a knockout of halting or at least dramatically slowing the spread of these tauopathies. Um,
Wow. And in those studies, so those mice will develop some learning and memory impairment as they get older. Have you looked at any behavior, or do you just look at the spreading of the pathology?
We are just starting to look at the behavior now. So one of our collaborators, who is still at Hopkins, she was interested in sleep abnormalities that occur with many neurodegenerative diseases, including Alzheimer's, and found some associations with tauopathies and sleep disturbances, and were able to improve sleep quality, duration, and quality with this small molecule. We are still looking at some of the behavioral improvements with um, inhibiting tauopathies. As you know, the amyloid, the I guess the cognitive impairments in most of these mouse models, unless you go with five mutations, can be somewhat mild.
That's
Um,
true. Yeah.
yeah, so we began with the APP-PS1 mouse. We saw very few effects on amyloid deposition. We saw, a minor reduction, but it wasn't great, and we didn't really see any behavioral improvement there. And we're just now starting to work on the 5X FAD model.
Did you,
Um,
so the 3X TGAD mice, we helped develop them.
mhm.
I don't even remember this, but yeah, sure you do. So you came to my lab at University of Kentucky almost right when maybe just a little before I made the decision to move to Baltimore to the National Institute on Aging. And then you moved to Baltimore when we moved the lab. And but what was I going to say? Yeah, at that time, no, it was when was it? It was right about that time. So I talked to Frank LaFurla,
Oh, I remember him. Yes.
right? And we have the presenilin 1 mutant knock-in mice.
Hmm.
Presenilin mutations are the major cause of early onset inherited Alzheimer's disease, and George Martin's lab had actually made the presenilin 1 mutant mice, and they were very interesting because they didn't develop any amyloid or tau pathology, and the reason is they don't have the human amyloid protein, and but they did have some interesting alterations. Anyway, Frank had this keen idea, and he did it. We sent him some of our mice, and so the idea was we want to have mice that express the presenilin 1 mutation.
Mm,
It's knock-in gene, which means the expression is under the control of all the normal transcription factors. It's not like a conventional transgenic where the gene expression is uncontrolled. It's just overexpressed.
mm,
Okay, so we had those mice, we sent them to him, and instead of crossbreeding them with, say, an amyloid precursor protein mutant mice, and then with the tau mutant mice, and it got to do all this breeding, he just got a female pregnant PS1 mutant knock-in mice, took out the 1 or 2 stage embryo, and injected with expression vectors for mutant APP and mutant tau. And then that individual mouse grew up with all 3 mutations. That's the 3XTGAD mice.
yeah.
Now they do develop cognitive impairment in our lab as they get old, uh, beyond a year of age, a year and a half, for example. Earlier on, not so much.
Yeah. And I think that is why we and a lot of other people have gone with the 5X model,
Yeah.
because you can start seeing impairments around 6 months of age.
Yeah.
I prefer fewer mutations, but of course, as you said, then you're looking at a year plus before you begin to see any obviously measurable cognitive
So
effects.
you have
Yeah.
these very exciting findings with these neutral sphingomyelinase inhibiting drugs in both models of HIV uh, and tauopathies, Alzheimer's, frontotemporal dementia, like who's the Die Hard actor? Um,
Um,
Bruce
Bruce
Willis.
Willis, correct.
Yeah, he's really in towards the end stage, I think, with frontotemporal dementia, which is pretty much purely tau. There's not necessarily any or negligible amyloid.
Yeah, yeah.
And so obviously you must have filed for patent protection. Uh,
We have a number of patents around the structure, um, um, secondary modifications, things where we're looking to have more of an extended release profile, and then some of the uses. And I think sort of what's interesting, you wouldn't think that there's any relationship between HIV and Alzheimer's. Um, and really, it seems to be that this generation of ceramide, which forms viruses, also forms extracellular vesicles. And the mechanism of virus release is the same as extracellular vesicle release. And the spread of this mutated form of tau, this hyperphosphorylated form, and other mutations, is spread in the same way that a virus spreads itself. It's through the generation of something that looks very much like a virus. It's about 100 nanometers, and it spreads it throughout the brain. So um, it was quite an interesting. Um, it's been an interesting area of research that's allowed us to
so
look
what's
at.
the plan then as far as clinical trials? Like so you get somehow so say you get patent protection. Usually you have to get at that point some company involved,
Yes,
or
yes,
that get interested and then put up money for initial safety studies, and then if that works, you go to some disease. So is that what you're going to target? HIV,
that
dementia, and
Actually, I think we're going to target Alzheimer's disease for
Okay.
a number of different reasons. There seems, if you're looking for venture interest, that's where you're going to
Well,
find
there's a
more.
lot more people with Alzheimer's disease.
Exactly. There's a lot more people with Alzheimer's disease. That number is going to continue to grow. And with HIV and antiretrovirals, if you keep the virus suppressed, people do okay. They don't die. They still develop cognitive impairments over time, but it's not as urgent, I don't think, as a target or
Yeah.
as favorable of a target to investors if you're looking at sort of
Yeah.
marketability. So we are at the stage now where our PK, which is the pharmacokinetics, as you take this drug or this small molecule orally, it has pretty decent PK. We would still need to dose, 3 times a day to sort of keep things in the IC50 range. So we are looking at longer acting preparations. I think once we get the secondary structures in place that allow the PK to look a little better, then I think we'll be able to move this into at least a phase one. At the
And
moment,
then, uh, but so you have given it to some humans?
well, no, not yet. At this
Just
moment,
just in animals.
just in animals. At this moment, we have tried mice, dog, and
Yeah. Yeah.
monkeys and
Yeah. Yeah.
primates. And the PK gets relatively worse as you go up the evolutionary chain. And for some reason, in monkeys, it is clearing extremely quickly. So now we are just starting some work with the Tulane Primate Center to look at some of these more modified versions of our small molecule that we hope will exhibit better PK in
So
higher
maybe
species.
maybe the
Yeah,
monkeys have some liver enzyme that's different or more active or
that's possible.
and
Whatever it is, it's metabolizing it faster than we would like.
so say in the rodents, let's go
Yeah.
with that. So you've shown that if you give it systemically to the rodents and then whatever, take out extracellular vesicles from the blood or take tissue that levels of you see any changes in levels of the ceramides that should change or
We
you have
we
an
do.
assay for the sphingomyelinase activity?
We do, and initially we had some concerns that because extracellular vesicle biogenesis is extremely important. It's as we said, it's a range of short. It's a mechanism of short and long range communication, and it's also garbage removal. So if you can't remove cellular products, metabolites that otherwise would be toxic to cells, ultimately it's not going to be good for any organism. And what we found is that the cells shift. They still produce extracellular vesicles. They're larger, but there's the same number of vesicles being produced. So they find a way around this one enzyme, and they find a way to get rid of the cellular garbage. So at least from a toxicity standpoint, we think that is the reason that we don't see toxicity. Um, the effective dose in both animal models, an Alzheimer's animal model and a humanized viral model, is about 10 milligrams per kilogram. If we go up to 100 milligrams per kilogram, approximated, oral dosing by putting it in chow, they start to lose weight. But then we lower the dose and they're fine again. But around 10 to 50 milligrams per kilogram, there doesn't seem to be any toxicity. And we think the reason for that is because the cell finds another way to remove what it needs to from the cell. It simply finds a way around this enzyme.
Yeah, interesting.
Yeah.
And there are a lot of other, there's a lot of scientists who are not neuroscientists or focused on neuroscience, whatever, cancer biologists, immunologists, etc. And these same lipids have been studied, are being studied a lot in those conditions. Would there be any applications of your drug to some of those other, like cancers or?
So, and in fact, so for ALS, one of the mutations that causes degeneration in the lumbar cord initially, right? That came from your group, if you remember, many, many years ago. one of the first things to go up, it was an SOD model, right?
Yeah.
A
Yeah.
superoxide dismutase
Yeah.
mutation model. One of the first things that goes up before you see any neuronal loss, or as you're seeing neuronal loss in that lumbar region of the cord, is ceramide.
Yeah.
For that particular disease, there seems to be some component. Um, Cancers, we haven't looked into too much. We haven't, but there is a woman named Niedergard, a Harvard scientist. She's worked on glioblastoma and numerous other types of cancers. Just a brilliant scientist. And she has found that a metastatic, so in order for cancer to spread, you have a group of cancerous cells and they spread to an area called a metastatic niche, right? It's some area that is favorable for these mutated cells to go and then grow. And the metastatic niche is formed by the release of extracellular vesicles from cancerous cells. Um, so it's something that we've wanted to do is to try this in glioblastoma, which, as you know, there really is not a good therapy in place for at the moment, but we have not quite gotten there. So there may be some utility in some cancer models, but I think it's too
Have
early.
you taught, Justin Lathian?
I do not.
Yeah. So Justin, he got his PhD in my lab. It was through the NIH Cambridge University program. So he got his PhD formally from Cambridge, but he did most of his research in my lab where he did some nice work on neural stem cells and the role of the extracellular matrix and integrin signaling and that. But he was interested in cancer and glioblastoma, and he's a full professor at Cleveland Clinic now, uh,
I'm going to look him up now.
working on. I did a podcast with him a couple of years ago. He's going great guns, and yeah, there's a really good, strong glioblastoma, both clinical and basic research group up there,
At
at
Cleveland
Cleveland.
Clinic.
Yeah,
I'm now going to look him up. I think his time in your lab was probably after I had already left.
yeah, it was.
Yeah, so I missed him, but no, thank you. I'm going to look him up. I need we need a model system to test this in. That's a glioblastoma. Yeah,
Yeah, cool.
yeah.
Uh, uh, let's see what else. Right, biomarkers. Right, so these ceramide levels change from various conditions, maybe with age itself, during aging itself, there may be sex differences. What about use of these ceramides or related things for biomarkers? That's a big
Yeah.
area now. Yeah,
That is something we have been dabbling with for years. It's absolutely clear in many neurodegenerative conditions, well, several neurodegenerative conditions, that ceramides do go up with the severity of the condition. Um, so you can, in our case, we were interested in using them as a surrogate measure. So, for instance, as you know, an initial trial is going to be 3 months or so long. If you're studying something like Alzheimer's disease, the chances of you seeing a noticeable change or improvement in cognition in those 3 months is not great. It doesn't mean that you don't have a good drug. It means you have a disease that has a much longer timeline
yeah,
than 3 months. So in our case, these are excellent surrogate measures for the effectiveness of our drugs to show that, in fact, we are hitting our target. We are reducing the levels of the product that we are interested in, um, even if we don't see a cognitive improvement within that time frame. So it's very useful for as a measure for things that are we target with a therapeutic. We have yet to figure out a lipid signature that we can assign to a specific disease. Our initial thoughts were that there may be different forms of ceramides and other sphingolipids that change very specifically with disease, and we're not quite there yet. So I think it's useful in combination with a number of other biomarkers, maybe to diagnose and to sort of understand disease, certainly as a surrogate measure if you're targeting some of these enzymes. But the specificity for a particular disease, I think, is not quite there yet.
Yeah.
Yeah.
Now, maybe a last question.
Yeah.
As you know, we did a lot of work on effects of caloric restriction, intermittent fasting, uh, on the brain and aging, disease models, and so on, and a little bit we did with exercise too. And right, once someone is cognitively impaired, there's already a lot of damage done. So ideally, one would want to prevent or reduce their risk from ever having cognitive impairment. And there's pretty good epidemiological data that uh, kind of a obesogenic lifestyle is not good for your brain as it ages. So not getting exercise, overeating, not exercising your nerve cells like you and I are doing now, uh, keeping intellectually engaged, socially engaged. And so how do any of these good environmental factors affect the sphingomyelin ceramide pathways? Has there, I don't know this, I haven't been following it. Is there any work on that? Because you'd think
Yeah.
maybe they do.
So, and we had someone approach us almost 10 years ago now, and it was a scientist in West Virginia who was conducting a fru-veg study, they called it, where they took adolescent aged children and put them on a diet that was very rich in fruits and vegetables and compared to a more Western diet, which has a much higher fat content. And they sent us samples from these adolescents prior to, and then I think it was 6 months after they had been on these diets. And the results were quite striking. Not only cholesterol and circulating triglycerides were lower, ceramides were lower. So the Western diet seems to produce, for reasons that we don't yet understand, higher levels of ceramides. And if you look at something that's more, in this case, a somewhat Mediterranean, or at least has a higher fruit and vegetable content and a higher fat content, yes, you can reduce those levels. So it does tie into diet, absolutely. One of the biggest one of the biggest ways that you can increase ceramides in your body are through meats. Meats and milks are very rich in ceramides and sphingolipids. Yeah.
Huh, yeah, interesting.
Yeah.
Okay, so how long have you been down in New Orleans now?
January will be a year, not even a year.
And how many people moved with you from Baltimore down there?
Initially two, my mass spectrometrist and then one postdoc. We're up to seven members in the lab now. We have one of which who is working with a collaborator at NCI, the National Cancer Institute, working on HIV though in Bethesda. So we have six.
Yeah, that's good.
Well,
Good sign.
We're finally getting to a point where the tissue culture is working. You know, you've moved. This is my first move experience. It takes a while, a frustratingly long time to get
You're
things
getting
operational.
contamination?
Oh, we did initially. That's it always happens, and then now it's good again. And I have to convince them that no matter you don't you can put on gloves, Matt. You can do whatever. That's not your problem.
Well,
You.
actually, I don't want to get on all this technical stuff in the podcast, but one of the big problems is that the airflow, and are there HEPA filters in the,
Mhm.
things coming in from outside?
hmm
Uh,
and you taught me if you're going to do, primary cell cultures from animal surgeries, you go in early in the morning before
yeah.
people start moving around.
Yeah, and
Uh,
that, by the way, thanks for getting up early, right? Because a little early.
6 a.m.
You're in San Diego, so that's 3 hours time difference. So yeah,
It
yeah,
is. I don't mind at all. It was a pleasure to see you, Mark.
yeah. Likewise. Well, enjoy the meeting. It's just you got a few more days there, I guess.
Couple more, couple more, and then it's back to Tulane and back to work. But
Okay.
it's been a fantastic meeting here, and it's really great to see you again.
My at neuroscience meeting, my back would always get sore. My lower back from standing all day, not necessarily moving much, like at the poster sessions, for example.
Mm.
And yeah, it's like 10 hours straight on the feet.
It
For
is long days, and certainly your brain gets tired, but the amount of information that is available at this meeting, as you know, is just incredible, right?
yeah. Okay,
Yeah.
Norm. I'll let you get going.
Thank you, Mark.
Yeah.
All right,
Appreciate
have a good
it.
day. Bye bye.
Bye.